Okay, today we shall discuss again on biofilm, processes. And one important point is that from next week, you shall face some quiz exams as well as midterm exam, first midterm, because lots of things already been discussed. So we need to have, I need to test you whether you are really following or not. So evaluation process is part of the learning. So we already discussed the difference between nanofilm or and biofilm, nanotechnology and biofilm technology. Uh, Biofilm, generally how biofilm is formed, the structure is formed, and what are the components present in the structure is important to know. For example, biofilm, I told you, especially bacteria, they form biofilm around the substrate. For example, this is the substrate, this circular green color one. So this substrate um, is a carrier. Carrier means a substrate, um, and like a biomass particle or like this. So this substrate, you see around the substrate, uh, the layers and lines indicating the biofilm. So here the composition of biofilms are shown. A carrier, this is the carrier. Then a B biofilm, that means the other around the carrier is the biofilm. And C is the fluid. Fluid means liquid because biofilm form and needs fluid. And then one cluster, there are lots of other structures. One cluster, a cluster, and a exopolysaccharide, two exopolysaccharide. So they use exopolysaccharide secrete from the cell to stick on the uh, carrier, that is the substrate, and then they form structure. And here you can see it is a bit perforated, not solid structure, so that oxygen can pass through, because they need oxygen for burning organic matter to get. So lots of pores are there. And cells at the base, they are inactive, and at the outside you see some cells are mobile also. So this is the like a house structure they develop. It is called a stylized scheme of biofilm system formed on spherical carrier. If carrier is a spherical, then in the liquid medium they form a biofilm like this. So you can uh, uh, easily one by one check the different components of biofilm structure and get the detailed information. Uh, there is a biofilm on the plane surface also. Plane surface, for example, a wall uh, uh, or drainage wall or like that. Plane surface, they also form biofilm. Uh, and this is the plane surface, a plane object, and this is the surface. So they form the biofilm in the similar manner. But here not all the side, only one side, and they form the biofilm. So similarly, pores and other things already there, and some cells are weakly connected, and some cells are mobile around the biofilm structure. In the beehive or wasp hive. You can see some are flying around the hive, and some are in the nest, staying in the nest, and they make the house like that way. Uh, biofilm structure. Biofilm structure, formation of biofilm structure on the frontier with liquid phase at different scales of liquid flow, linear velocity is involved. Uh, for example, low velocity, middle velocity, high velocity. Velocity in the different part is different, and uh, in some states, uh, you know, the biofilm becomes the stable. So in the biofilm, liquid always circulate, and so that nutrient can also circulate. And this is why you see there are pore spaces, because uh, biofilm forming, for example, bacteria, they use different enzyme to break down the materials, nutrient contained materials in the carrier to get the nutrition, and share with others. So it is always circulating in different ionic form. So there are some velocities in the biofilm. That means it is a dynamic structure. Here, biofilm structures formed, various states of fixed cells at the beginning of biofilm formation. At the beginning of biofilm formation, you see, for example, some active cells, and this is the carrier. So they aggregate and then work together. And here you see gradually, and they are active cells. But when exopolysaccharide structure is formed, some cells inside the exopolysaccharide, they remain inactive, but they hold the biofilm structure. And here you see the strongly bounded microbial cells due to red color means some polysaccharides tightly bound and attached to the carrier, and some are weakly linked. So this is the way they form the biofilm structure. So dynamics of biofilm formation and functioning. Uh, Biofilm formation and functioning is carried out in open system. It is always open system, no mass and heat transport limitation in the liquid phase. So heat and mass can transport, and there is no limitation. No limitation regarding the constant cell concentration in the liquid phase. So how many cell can be constant at the base, or how many would be mobile phase, no limitation. So it is a bit flexible. There are, seven major stages of biofilm formation. Let me tell you the story, how they start, and how the biofilm, structure is completed, and how they break down the biofilm structure. So stage one uh, is adaptation of cells to the carrier surface. For example, this is a carrier, and this is a carrier surface. So these are the biofilm forming cells. So they will first visit and inspect the environment. If environment is good for them, they will use different signaling system, sensor system, to understand whether this place is hospitable for them or not, whether safe for them or not. They will also explore, biofilm forming organism also explore whether this substrate or carrier is good for future nutrition and shelter. So you can see all the cells are not attached closely. They are moving and inspecting and sensing and inspecting the uh, uh, you know area or carrier, as well as the areas, micro area called substrate, whether they will get enough oxygen, enough safe fluid or nutrient or like that. So at the stage one, you can see biofilm like that. Some a few cells adhered, and all are swimming cell. Uh, so this is the called cell adaptation and single cell attached. Single cell, some single cell attached. This is the stage one. So in stage one, cell adaptation taken place, swimming cell, few cell adapted, settled there, and swimming cells reproduction going on, conversion of substrates by swimming cells to produce biologically active compounds. Conversion means they use different enzyme to break down the substrate and check whether they will get enough energy and environment for developing a biofilm or not. Exopolysaccharides on surface, cell surface, need for their future fixation. So they also release exopolysaccharide for future fixation on the surface of the carrier. Specific compounds for carrier surface preparation for cells fixation byproducts. So only sporadic single attached cell you can find the fixed. So most of the cells are swimming, and swimming cells, they also do some function, and at a cell they also do some function and test the environment whether adaptable or not, or hospitable or not. Physicochemical phenomena at this stage: adsorption of nutrients from the liquid phase. That means liquid phase must contain nutrient, otherwise how can they multiply and grow. Adsorption of specific compounds excreted by the cells, adsorption of cells on the carrier surface. That means spontaneous fixation, cell fixation on base of electrostatic interactions. So these are the biological phenomena taken place during the first stage of biofilm formation. What are the results? Carrier surface prepared for cell fixation. First adhered cells, needed products. That means they need some products for the adherence as well as their nutrient. Prepared surface for cell surface for fixation, so that other cell can fix. So they prepare the carrier surface so that other can fix there. Biofilm system state, homogeneous mechanism of bio conversion. So no biofilm formation, isolated cases of attached cells, domination of swimming cells productivity. So these are the phenomena state. Only few cells are attached, but all are swimming. Necessity of intervention into cell organization. In the case of processing technologies, artificial carrier surface modification for acceleration of the cell fixation. So necessity of intervention into cell self-organization. So they need some intervention for self or organization. So in the case of processing technologies, artificial carrier surface modification for acceleration of cell fixation. That means they modify the surface by using different biomolecules so that other cell can easily fix on it. That means on the surface of carrier. In the case of protective or impermissible technologies, deliberated carrier surface treatment to avoid biofilm formation. So there are lots of the job initially they do for forming the biofilm on the carrier surface. So you may ask me a question, how long it would take? It would not take so long time, because in case of bacteria, they can multiply very fast within 24 hours, and they can do the job very fastly. Let us see the stage 2. Stage 2, formation of cell monolayer. Here, in the previous one, only one or two scattered cell were attached. But here you see a monolayer, almost a single cell layer is formed. Here you see around the cell, there are some, envelope-like structure. Envelope means exopolysaccharide secreted from the cell. Exopolysaccharide, they are sticker like, sticky substances, so that they, enveloped the cells, so that they can easily settle and stay stably on the surface. So formation of cell monolayer is the, characteristic feature of stage 2. And formation of um, cell monolayer is uh, facilitated by the use of exopolysaccharides released from the biofilm forming cells. So these are the biological phenomena. Cell adaptation, obviously swimming cells. There are two types of cells, swimming cells and fixed cells, because already monolayer is formed. So swimming cells, the phenomena are the like previous one, and fixed cells, beginning of the fixed cells reproduction on the carrier surface, isolated case, and conversion of substrates by fixed cells, that is products, biologically active compounds they produce. Some of the compounds are antimicrobial to other organisms, so that other organisms cannot invade their chosen biofilm forming carrier surface. So some defense molecules also they can produce. They can produce some signaling molecule like isohomoserine, lectins, that means quorum sensing, signaling substances, the substances they use to communicate with each other, how fast the biofilm should be formed, and they communicate like that way. Obviously, substrate for biofilm formation and its structuring is facilitated by the application of exopolysaccharides, precipitated salts, by the use, secreted from the biofilm forming bacteria or organisms. And there are some byproducts also. Physicochemical phenomena in the first stage, absorption of nutrients from the liquid phase, because otherwise they cannot multiply. Absorption of specific compounds excreted by cells. Absorption of cells. Absorption means absorbed on the surface of the biofilm. So these are the basic biological phenomena happen during the stage 2. So what are the results? Envelopment of cells with substances for biofilm formation, mainly EPS, exopolysaccharides, for making the single or monolayer of the cell on the surface of the carrier. First cell configuration on the carrier, cell monolayer formation, fixed cell reproduction, and needed products. And they needed products for the reproduction. Biofilm system state, homogeneous or heterogeneous mechanism here, domination of swimming cells, productivity, and fixed cells start to precipitate in bioconversion. Bioconversion means conversion of the carrier, because they use different enzyme for the bioconversion. and intervention into self-organization. In processing technologies, no need. In protective technologies, mechanical means high shear stress. That means if you want to promote this stage, what you need to do, because this type of biofilm are also used in the industry. So in the processing technology at this stage, no need. In protective technologies, mechanical means high shear stress around carrier surface, scraping off biofilm form, biocides, introduction in the liquid phase is necessary. Otherwise, that means if you want to protect the surface from the biofilm forming bacteria, then you need to use biocides, so that or some other means increase the shear stress, so that organism cannot form biofilm and damage your surface. So these are the results. Similarly, stage 3 biofilm structure, you see gradually in the previous stage there were monolayer. Here you can see bilayer thickness is increased. First critical thickness you can see, it is called delta 1 CR, first critical thickness. This part 0 to this part has already developed. Swimming cells number decreased. So start of the stage monolayer, but end of the stage polycell layer, first critical biofilm thickness developed. So you can easily understand the biological phenomena like the previously I discussed. Results also similar. So you can go through, and if you have any problem, then you can ask me. Let me show you the stage 4. In case of stage 4, you see second critical thickness is formed, and here you see biofilm system, how it is developed. First critical biofilm thickness, delta 1 CR and delta 2 CR is developed. Delta 1 CR is up to like that. So biofilm is gradually increasing, and please go through this part. But at stage 5, uncontrolled and unstable biofilm growth, you see, not controlled. In the stage 4, there were two critical thickness, one critical stage 1, critical stage 2, but here you can see uncontrolled, the surface is uneven. That means uncontrolled and unstable biofilm growth. Cavities formation, some cavity, here you see cavity is formed, and this is characteristics for stage 5, cavities formation. And please go through this part. And stage 6, you can see the how to say biofilm destruction is taken place. Third critical biofilm thickness is also formed, but biofilm, some part of the biofilm is destroyed, that means separated from the carrier. Here big cavities is formed, few swimming cells are there. So detachment of parts of biofilm, detachment from the original carrier is taken place. And then last stage, restart of new biofilm formation, simultaneous realization of all stages. That is restart of new biofilm formation. So again, biofilm is forming, and cavities formation still remain. Detachment of parts of biofilm volume is remain, but they are attempted to restart a new biofilm formation. So biofilm formation on the carrier is a dynamic process, and first adaptation and inspection, and then gradually monolayer, and then critical layer 2, and then gradually when the nutrient as well as bioconversion is exhausted, and then biofilm breakdown is taken place. And after breakdown, the cells move to even new place. to make the new biofilm considering the environment. So biofilm formation, for biomass conversion. Uh, um, in biomass conversion and other technologies, biofilm formation, you have to understand, is a dynamic process. Like in the composting process, composting process, the biomass you use and the microorganism participates, they are changed. Different microorganism in different stages, they participates and break down the biomasses, and finally release the gas and very finer particles of nutrients. We can use the effluent as fertilizer, and we can use the gas as energy. So biofilm formation, 7 stages are like this, and it is really very interesting. And application in biofilm reactor design and development. In case of biofilm reactor design and development, all the stages you have to understand their principle and performance. You need to test outside, and then you can make a reactor like this. And obviously air supply is necessary so that they can be stable, and nutrient supply also necessary so that they can function for a longer time. The 7 stages I showed, they are basically happen in the natural environment. So this is all for today, and maybe in next class we shall discuss more detail about biofilm reactor design and development, because biofilm technology is a powerful technology in environmental biotechnology, and it can be used for many purposes, for making the valuable secondary metabolites, for making the enzymes, or for uh, uh, cleaning the toxic hazardous materials, or conversion of the biochemicals, toxic biochemicals to other one, or even making a new bioactive compounds. So this is all for today's lecture. Let us discuss a bit if you have any question. Please ask a question. Unmute yourself and ask a question.
Sir, Assalamu alaikum.
Walaikum.
Sir, is there any special factor which triggers the growth of biofilms, and what it is or what they are, sir?
Yes, there are lots of uh, um, factors involved in triggering biofilm. Uh, the most important factor is the carrier must be suitable for the biofilm forming microorganism like bacteria. As, because bacteria has the sensing system, they first sense whether carrier is suitable or not. Other thing is that the around the carrier, the liquid, uh, either it is water or any other liquid, must be contain nutrients. So nutrient is a factor. So if a nutrient poor condition, biofilm formation also be not good, because for multiplication of the bacteria and production of different exopolysaccharides or bioactive compounds, they need the energy. They need to multiply their cells. So other thing, pH of the environment, pH of the liquid medium, uh, obviously temperature, pressure, lots of factors, availability of the oxygen, that means aeration is also important. So lots of factors are involved in facilitating the biofilm. In natural system, for example, in a pond or a lake, the bacteria which are in the biofilm, they are on the surface of different, carrier or object. Bacteria you cannot see, and even their biofilm you cannot see in the naked eye in most cases. But they are there. So depending on the factors that I mentioned, the biofilm structure, their dynamics, and their activities are regulated. So when we want to use biofilm forming microorganism uh, in an industrial process, we have to thoroughly investigate all the factors that facilitate better biofilm formation and function of those cells. Then you can use those microorganisms or that microorganisms for the industrial application. So you have to do some experiment at the basic level, basic study, even genomic analysis, as well as other microbiological study. In the laboratory condition, you need to do a pilot, experiment to see whether, if you ensure all the factors that is loved by the biofilm forming microorganism, uh, what is the outcome. So you can, in small scale, you can assess their performances and change the different factors to see whether you can increase the performances or not. For example, if you want to produce a drug, secondary metabolites or bioactive compounds by some biofilm forming bacteria. In that case, if you offer them, very good and hospitable environment, including the high nutrition, they will not produce enough secondary metabolites. So what you need to do, you need to, uh, add some stress so that as defense they produce the secondary metabolites. So lots of basic and microbiological studies are needed to utilize the microorganism to understand the condition of their gene expression for the particular job is related to. Then you can utilize them in the best way. Okay.
Thank you, sir. Ji, sir.
Welcome. Any other?
Sir, Assalamualaikum, sir.
Sumaya, bolo.
Yes, sir. Sir, in the biofilm system state slide, we see that homogeneous modification bioconversion. Sir, what does it mean? Sir, homogeneous bioconversion.
Yes, it is actually when biofilm is formed heterogeneous. Heterogeneous means not some cells are active, some cells are inactive, some are uh, all are not playing the same role. Like in the community, all are not playing the same role. Uh, uh, so complex heterogeneous activities, heterogeneous roles, heterogeneous cells are involved. Some cells at the base of the biofilm, they are inactive. They are holding the structure. But the swimming cells, they play a huge job to, convert the nutrients, multiply their cell, produce the exopolysaccharides, and support the biofilm. So it is really a dynamic process, and basically a heterogeneous population of the microorganisms are there. Any question?
Thank you,
আসসালামু
sir.
আলাইকুম স্যার।
Jahid?
স্যার আমি একটু সিক আজকে স্যার, আপনার ক্লাসে তেমন মনোযোগ দিতে পারি নাই স্যার। তবে স্যার আমার একটা কোশ্চেন মানে কিউরিওসিটি আছে স্যার বায়োফিল্ম সম্পর্কে। স্যার যে মানে যদি কোনো একটা অর্গানিজম স্যার বায়োফিল্ম প্রডিউস করে, সেটা কি স্যার মানে এনভায়রনমেন্ট থেকে বিচ্ছিন্ন হয়ে যায়, মানে অ্যানাএরোবিক হয়ে যায়, নাকি সেটা পারমিয়েবল বা সিলেক্টিভলি পারমিয়েবল থাকে স্যার ওখানে বলতে।
I told you 99% of the bacteria, for example, they remain as biofilm form. Even all the organisms, they remain as biofilm form. That means form the structure and colony and like social being, they stay. But they never, detached from the environment. Environment is a complex, term. It includes everything: biofilm, non-biofilm, biotic, abiotic, all the factors and all, organic, inorganic, living, non-living, in a given place, all together we can call environment. So environment is includes obviously biofilm also. They should not be detached from the environment. Any question?
No, sir. Okay, sir.
And then thank you very much, and please concentrate yourself for preparing the review that I suggested in a group work. So what you need to do within the weekend, you need to shape a structure, and next week what you can do, you can show me the first draft, because it needs time and several phases like biofilm. First, you need to gather the information and organize it and make a first draft. Uh, um, so what you can do, you can divide the job among yourself, and then combine the part job and make the whole draft, and someone needs to edit and make a single story from different parts of the story. And the first draft, if you can show me, even you can present through the Zoom meeting, and then I can make some comment how you can proceed, and for making a finest review. Obviously, it should be a good story, and it should add some information to the science, and it should cover all the information available, latest information available, and obviously it would be a good learning for you, and you can be confident enough that what is going on all over the world regarding the biosafety of uh, genetically engineered and gene edited crops different countries. And you can also add your thought what should be what in some country there is no reason that they are not accepting the high tech. In some reason, they do not need the technology. For example, European Union, they have their population is declining in all the countries, so they have no need to get more food. So they are going back to organic, but in the Asian as well as African countries, in the developing countries, population is increasing faster, land is shrinking, and you need, advanced technology for uh, ensuring food and nutritional security of the growing people. And you must also have understanding that in the sustainable development goals that set by United Nations organization for all the countries, which should be achieved by the year 2030. 17 goals. The second goal is zero hunger. So no hungry people should be exist by the year 2030. So very big goal, and to achieve that goal, you have to understand by the 2030 how many new people our population will increase in the developing country, and how much more food is necessary for them, and whether existing technology can meet up the growing demand of the food or not. But it is now clear that the currently used technologies are not, uh, you know, uh, sufficient to produce, more food for the more people. So please be critical and think as an agriculturist as well as an intelligent citizen of this earth, and in a broader sense, and try to share your thought in the review. So your review should not be just a copy of others one. You can take the idea from other reviews, as well as you can borrow some sentences or style also, but you are not allowed to just copy and paste, because it is plagiarism. You must need to rephrase your story and make your own story depending on your own structure. And You can, if you use something from other literature, you must cite it. And in case of figure, diagram, table, you need to make of your own way, and redraw or change it, so that uh, no problem would be associated with the copyright, because we are not allowed to use others image, others figure, or others text directly to our own story. So this would be this assignment not only give you a better understanding of the subject, it will improve your way of thinking, as well as uh, your understanding to deal with science. And it is very important at this budding stage of your scientific career. Thank you very much. Any more question?
No, sir.
No, sir. Sir, assalamu alaikum.
Okay. Thank you so much. Uh, have a nice day.